Today I will be teaching you about the guidance of pioneer axons and more contextually about the way that I use genetics in my research to study axon guidance. So the goals for this lecture is, first and foremost, to give you a firm ground and an intro into using genetics to study neurodevelopment. And also to give you a taste of graduate level research and research life. I'm also hopefully going to be able to connect genetics to behavior and how genetics can affect neural function, essentially. And also I enjoy talking about my research. So I'm a little selfishly going to enjoy myself and indulge in these stories, because they really are stories to me. They're building this world and this understanding of this topic that I'm so passionate about. So as you read, you'll notice that there are colors. I color code a lot of my words in general, but the things that I want you to keep an eye out for are this blue text, which is just important concepts to be aware of. Just to help you kind of skim these slides. There's also black bolded text, which are vocab words. I will be writing a couple exam questions for your final. So these words that are black and bold are words that you're responsible for and should kind of understand what they mean. And the orange text are questions that I would like you to think about. Either they're going to be little mini quiz questions with multiple choice, or just questions that I had to think about as a researcher. So, before we get started for realsies, let's talk context to give you some real framework for what we're going to be doing. This is a neuron. It's a baby neuron. Here's its cell body and its axon, and it has a developing growth cone. And as this growth cone develops, it's early. There aren't any other axons in the neighborhood, so it's going to be using attractive and repulsive cues in the environment, either emitted from a source, creating a gradient, or in a contact-dependent way, where these cues are stuck to a substrate. Later axons can then follow this pre-existing path that our early axon laid. This is how C. elegans builds its ventral nerve cord, which is analogous to your spinal cord. This is a schematic of the worm. There's its head, here's its tail, and early on, the AVG, which is this cell here that I want you to pay attention to. Here, let's circle it. Let's see if that works. AVG here is going to be our pioneer, and it's going to extend early on. And then later, these followers, including especially these interneurons in green, are going to follow that AVG. And this pioneer-follower relationship is highly conserved. This is the basis of our olfactory bulb formation, as well as the growth of our corpus callosum. And there are pioneer-follower disorders that result in the olfactory bulb not developing, causing a lack of the sense of smell. It's very interesting. And I just want to show you a little video here of AVG. So this is our worm, our embryo in its eggshell. And the head is developing up here, and you'll see those neurons developing. Now, this worm is expressing a green fluorescent protein, but it's blue, a blue fluorescent protein, in all of the cells that have a neuronal identity. So all these blue cells are neurons. And here's the brain developing, and I'm gonna pause it right here because this guy is our AVG cell. This is that first extending axon. This is its growth cone, and it's going to extend down here where the future ventral nerve cord is. And note there's no other neurons in the neighborhood. This is absolutely the first thing extending. And now, I'm going to pause it. You see how it started to twitch a little bit? That's because these cells are coming online. As they turn blue, these are our motor neurons. And as they turn blue, they are extending axons to the surrounding muscle, innervating those muscles pretty much at random, which is why it starts twitching. But at this time, we're going to start seeing some of these interneurons also extending axons down the ventral nerve cord, which is here, and you're going to see that getting brighter and brighter. But isn't that lovely, that AVG growth cone? My goodness, it's pretty. All right, so there we go. Aha! See how bright that is now? Here is our bright and shiny ventral nerve cord. And you can see it's brighter up here than it is down here because the interneurons are on their way. So fun, look at it twitch. Ah, okay, I'm cool, I got this. So here's an unnecessarily confusing schematic of the motor system, which is what you just saw growing. I study the pioneer-follower relationship of this motor system, which happens in the right ventral nerve cord. So if we go back here, you can see that AVG extends in just the right ventral nerve cord on the right side here. And then all of these other axons extend in the right also. So when we're in this motor system, that follower-pioneer relationship is all happening in the right ventral nerve cord. And in red here are the command interneurons. Their axons kind of facilitate the motor system. These command interneurons communicate information from the sensory neurons to the motor neurons. And they also communicate with each other and regulate motor response, which is a very important process. And all of those synapses happen en passant, which means in passing. So each of these axons needs to be in physical contact with each other in order to successfully orchestrate motor response. So if we have defects in this follower-pioneer relationship, then not all of the axons are going to be in the right ventral nerve cord, and because of that, they're not all going to be together, so they can't talk to each other effectively. Richard Durbin used a laser to kill the AVG cell, that tiny blue cell, at the tiny blue cell stage. When it's in the egg still. They killed that cell before it sent its axon down, even before it started to get blue, which resulted in some very strange worms with very disorganized ventral nerve cords that Richard Durbin in the 80s was able to capture via electron micrographs. Those pictures are very confusing and ugly. Informative, but ugly. My advisor, Harold Hutter, for his PhD, did this, which is absolutely beautiful. He did basically the same experiment that Richard Durbin did, ablating, killing that AVG cell. But instead of doing the electron micrographs, he used fluorescent markers. So here, the interneurons are in green. We've been seeing them in red in the past, and we'll see them in red in the future. Interneurons are red, generally. But in this specific image, they're green. And here we see that in the normal worm, all of the interneurons are together on the right side. In the AVG eliminated worm, we have those interneuron axons now on the wrong side, on the left side. And you can see them crossing the midline, which isn't supposed to happen either. And there's also some motor neurons crossing the midline. Both the blue and the red are motor neurons. So, with all this in mind, let's have a little quiz, because quizzes are fun! Which of these four statements is correct? We just saw command interneurons in green, now they're in red. AVG is green, and that's going to be true for the rest of the talk. So keep that in mind. AVG green, command interneurons red. Okay, so which of these four statements is correct? A, AVG axons follow earlier extending axons to find their way. B, command interneurons navigate using primarily environmental cues. C, AVG is required for asymmetrical ventral nerve cord development. Or D, none of the above. Pause, think about it, scroll back, scroll forward, come back. Are you back? Let's do this. It's C, the answer is C. AVG is required for asymmetrical ventral nerve cord development. Asymmetrical ventral nerve cord development is the ideal. It's the default. We want it to be asymmetrical because then it's all on the right and not on the left. This is the not good scenario, in which some is on the right and some is on the left. You see how there's some symmetry, there's some on the other side. The interneurons are separated. We want them together. Okay, so AVG is required for the asymmetrical ventral nerve cord development. Next question. If AVG is missing, command interneuron axons A are more likely to cross the midline, B are less likely to cross the midline, C won't extend into the ventral nerve cord at all, or D will rely more heavily on environmental cues to find the correct path. Give it a pause. Come back. Welcome back. The answer is A. They are more likely to cross the midline. So remember, the command interneurons are following AVG, and AVG is keeping them all together in the right. They're not supposed to cross to the left, but when AVG is eliminated, we see some crossing over events of these things crossing to the left ventral nerve cord. Cool. Next question. To determine what cues AVG axons use to find their path, we should A, look at the effects of command interneuron ablations. So using a laser to kill some command interneurons. B, look for mutants with bad pioneer-follower relationships. C, look for mutants with abnormal AVG axon paths. Or D, all of these approaches would work. So pause it. 1, 2, 3, and you're back. C, we're going to look for mutants with abnormal AVG axon paths. And when I say mutants, a mutant is something where there's been a gene messed up. That's a mutation. So when there's a gene messed up and the AVG axon is messed up, we're going to presume that that gene was important for that axon path. Last question. To find a gene involved in AVG axon guidance, we, A, remove genes for likely guidance cues and then look for AVG defects. B, remove genes known to affect command interneuron axons and then look for AVG defects in those mutants. C, cause a bunch of random mutations and then look among them for the AVG axon guidance defects. Or D, all of those would work. Okay, pause it, give it a think, give it a look, come back. And we're back. All of those approaches would work, and we use all of them. I'm going to talk about them all in the next slide. We go from easiest to hardest because researchers are lazy. So, how does AVG find its way? This is my research question. This is the basic premise of all of the research that I do. The premise is that if a gene is important for axon guidance, knocking it out will cause axon guidance defects. And by knocking it out, I mean causing a mutation in that gene, or causing a null allele or a deletion allele in that gene. And that will result in messing up the process that that gene was in control of and helping before. Without it, that's not going to be helpful. It's not going to be able to do its job. And we're going to see the effects of it not showing up for work, essentially. So that first easiest option, knock out likely candidates, something we did, and it didn't work. None of those likely candidates seem to affect AVG axon guidance. Here's a little smattering of them. You might recognize some. There's netrins and slits and ephrins and semaphorins and TGF-beta. And none of them have really any significant AVG defects going on. So the next easiest thing seemed to be knock out genes affecting command interneuron axon guidance. However, we have the opposite problem here. There have already been documented so many genes that affect command interneuron axon guidance. Like, so many genes that affect command interneuron axon guidance. Which leaves us with a genetic screen. Randomly destroy genes. We can do this with radiation or with liquid mutagen like EMS. And then we go through the offspring and find mutants that have defects by screening it. And then we sequence those mutants and map them. And then we figure out what the gene does. And we'll go through all those steps together. But first, let's talk about what we're looking for when we look through those mutants to try to find stuff where AVG is wrong. We're looking for this. So here's our AVG cell body right here, and there's its axon. And here's a lovely stop phenotype. Here's a noping out and leaving the ventral nerve cord phenotype. Here's a wandering off in a loop phenotype. And then our midline crossing defect where it's on the right, and then it's on the left, and then it's on the right. We used these to screen for mutants, and we did find one. We found one. We named it PLR1. This was the work of Jafar Bhatt. And PLR1 has two predominant defects. There's a premature stop defect, and there's a polarity reversal defect, where the axon goes into the head and kind of loops around and gets tangled, instead of going down the ventral nerve cord. So, when you look at this, what do you think PLR1's role in AVG axon guidance is? Does it induce axon extension? Keep axons from crossing the midline? Direct axons towards the tail? Or does it cause premature stopping and polarity reversal, and that's all we can see? So pause, think about it, come back, we're back. It directs the axon towards the tail. And I made a big point of putting that D in there to emphasize that PLR1 does not cause these defects. PLR1 is the gene name. The mutant allele causes these defects. So essentially, PLR1 prevents these defects. And if you remove PLR1, if you knock it out, if you mutate it, then you're going to see these defects. Mutating this gene causes these defects. The gene itself prevents them. Important distinction. So, PLR1 directs the AVG axon growth cone towards the tail, and Jafar was able to figure out that it actually does so via the Wnt gradient, that anterior-posterior gradient of Wnt activity. Remember that? PLR1 allows AVG to be receptive to that gradient. Cool, huh? So, why didn't we find more? Usually we find like 40 of these in a genetic screen. We only found one. That's weird. Why? Well, redundancy. Different factors doing basically the same thing, so that if you knock one out, the others will pick up the slack. The analogy I use for this is a spider. A normal, healthy, wild-type spider has 8 legs. But if horrible things happen to this spider by nasty people, and it ends up with only 5 or 4 legs, it's gonna be okay. It only really needs 5 or 4 legs. There are safeguards in place because legs are important. However, once it's down to 4 legs and you take another one off, it can barely walk. And if it's only got 2 legs left, it can't do anything. It's gonna die. So, when we're working with genetics in this context, in order to identify any active pathways or factors that are important in this process, we're going to have to take out several factors at once. And then we'll be able to see changes caused by removing additional extra factors one at a time. So, this brings us to nitrogen. Nitrogen is a part of the basal lamina. Basal lamina is an important developmental thing. It's in every gastrulated animal. It runs between the mesoderm and the ectoderm, where the neurons go. And it's been hypothesized as the substrate along which axons grow, because it's very easy to stick things to the basal lamina. It's made up of a bunch of different very sticky things that associate with other things, essentially. So this might be a substrate for signaling molecules. So if you take it out, all of the signaling molecules that are stuck to the basal lamina won't be there anymore, and it's going to make it harder for the neurons to grow. Without nitrogen, mice are very sick. Humans are dead, but C. elegans are fine, except that they have midline crossing defects. They have 12% midline crossing defects. Woo! Very exciting. So our hypothesis is that nitrogen is the substrate to which many legs are attached. Without it, we'll more clearly be able to see the effects of disrupting the remaining legs. How will we test this hypothesis? How will we disrupt those remaining legs? A, look at nitrogen mutants. B, run another genetic screen, but in nitrogen mutants. Or C, add candidate genes to nitrogen mutants and try to prevent defects. Pause it. We're back. Okay, cool. We're going to run another genetic screen. And again, C is something I would like to emphasize as not true. These mutants have no nitrogen, but they have all of the other genes. So if you add extra copies of those genes that they already have healthy versions of, not going to prevent stuff or cause stuff. Well, it might cause stuff because of dosage stuff. Don't worry about that. Anyways, we're going to run a genetic screen. Yeah. This is an enhancement screen because we're enhancing a pre-existing phenotype, or trying to. We start with worms that don't have nitrogen, and all of the other worms we will work with won't have nitrogen. This will all be nitrogen null, everything. We're going to mutagenize it, and then we're going to screen the F2s. And we got plenty! These are just 16 favorites to represent the fact that we got plenty. Do not try to memorize anything in this presentation that has a number. I promise you will not have to. Anything that has numbers, you don't have to learn. Next step. What are they? What's going on? So, each of these strains has many mutations. These alleles are just one of many in each of their strains. And only one of those mutations is causing our phenotype. And the first step is to isolate the mutation. And we do this by back-crossing. Blue is going to represent mutagenized DNA. So DNA that has been mutated by our mutagen. And for each mutant that we're working with, many genes have been mutagenized, evenly distributed throughout the chromosome. Each of these it represents a full set of chromosome. So because they're diploid, there's two, essentially. And so this one with the M here has a mutant phenotype. If you cross it with a control phenotype, then the F1 offspring is going to be heterozygous for all of these mutant alleles. Then we're going to back cross it. We're going to cross it back to the control parent. And we're going to do that again. And then we're going to take this worm and self-fertilize it, allow it to fertilize itself, resulting in homozygotes of mutations from our mutagen process. Some of these homozygotes will have mutations that cause our phenotype, and those will have the phenotype. So we take the offspring of the F2 and we screen for individuals that show the phenotype. Now that we've found an individual, it's been back-crossed, it shows our phenotype. It has thousands of genes on each of 6 chromosomes. And we need to know where this gene is so that we can confirm its genetic identity and figure out how it does what it does and why it's important for AVG guidance, AVG axon guidance. So, how are we going to do that? How can we tell where our mutation is? A, it's the blue part. See? Right there. You can just see it. B, we need to map it, whatever mapping it means. C, we need to sequence it so that we can see where the blue part is. So, pause it, think about it, scroll back, come back. We're back. There. It's C. We need to sequence it. It used to be that we would map it first and then sequence. However, sequencing is much cheaper now, and mapping is incredibly time-consuming. So, we sequence it. Specifically, we pay a company to sequence it. We send them the DNA, and then they send us a list of locations where our sample is different from wild type. Here are the sequencing results for a specific one, HD145. Each dot is a location on the genome that is different from wild type, presumably because of the mutagenization process that it's gone through. And because of the outcrossing, it's no longer completely covered in mutations. We're going to see fewer mutations because some of this has been swapped out with wild type DNA. We essentially selected for the mutation that causes our phenotype and against others. So one of these dots causes our phenotype. We know this because the worm that we sent for sequencing has our phenotype. So what chromosome do you think HD145 is on? And keep in mind, these are the 6 chromosomes. The 6th is chromosome X. It's our sex chromosome. So that's why those are there. And these are the dots, mutations on each chromosome. Okay, so pause it. Come back. It's X. It's on X. And it's on X because that's where most of the mutations are. We selected in our back-crossing process for the mutation that causes the phenotype, but we're also incidentally selecting for that whole region of the chromosome and that chromosome itself. So we're going to see more mutations in the region where the phenotype-causing mutation is located. So, my face is about to go away. I'm so sorry. This is because we're going to talk about something that crowds the slide a little bit. We're going to talk about mapping. Here it is. Where on chromosome X? Whoa! Giant object. So, this is how we map. It's a lot like how 23andMe works, where worms that developed in different regions will have unique single nucleotide polymorphisms, or SNPs, which is bolded, and that's a vocab word. All of our worms that we study come from Bristol, because the first worm that we started with was pulled by a guy who was working in Bristol. So we call them Bristol worms. And when we map, we compare them to Hawaiian worms. When you compare Bristol and Hawaiian, there are a bunch of single nucleotides that are different between Bristol and Hawaiian, fairly evenly distributed throughout the genome. They are so numerous and frequent that you can find a bunch of them by using specific enzymes. And so, through a process of seeing which regions can be digested by various enzymes, you can essentially color in the chromosomes and figure out what areas are Bristol originated and which are Hawaiian originated. HD145, our mutation that we're talking about right now, happened in a Bristol background. So this red dot is our HD145 mutation. And it's surrounded by blue, which means that whenever we find a red dot, it's going to be flanked by blue. Very important. And if we map it with our orange Hawaiian, then our F1 progeny is going to be heterozygous for every single one of those SNPs. And when it self-fertilizes, it will undergo recombination, resulting in randomly mixed blue and orange, essentially. So, for chromosomes where our gene is not located, it's going to be evenly distributed. Where chromosomes where our gene is located, like chromosome X, for instance, and when we select for a phenotype, and we look when we color things in blue and orange, depending on which enzymes digest things where, we're going to see a consistently blue spot. And that consistently blue spot is where our mutation is. So we're essentially ruling out sections of the genome where a worm can have a Hawaiian SNP and still show our mutant phenotype. So we did that, and we mapped our HD145 to a completely unknown gene. So what do we do? How do we figure out what this gene does? Hi, I'm back. New background, so sorry about that. My computer died, the sun went down, everything is complicated. Here we are in my kitchen. Look, my plants, aren't they sweet? I love them. How do we figure out what this gene does? So there are 3 ways to confirm an allele's identity. What gene that allele is a mutant version of, essentially. We're going to talk about all 3 of them. The first is complementation testing, which is comparing a known mutant where a candidate gene has been mutated, comparing that to your mutant and seeing if they're the same gene. The second is a rescue experiment where you inject healthy DNA of the candidate gene to see if it rescues the mutant phenotype. And the third is pathway confirmation, essentially seeing if your mutation acts as if your candidate gene has been mutated in the context of predictive genetic interactions. We'll talk about all 3 of these. We're going to do all 3. I do all 3. But I work in order from easiest to hardest. So, starting with complementation testing, we're going to order a worm off the internet. It comes in a little manila envelope, very exciting. We're going to order this worm off the internet that has a mutation in that region that we think our mutant allele is located. So this is a different allele, but of what we think might be the same gene. So here in orange, let's say, is our mutation, and in purple is our known mutant. So this is a mutant in a candidate gene that we ordered off the internet. We're going to mate the 2 of them together and look at the first generation offspring. And if both mutants affect the same gene, then this will look just like this and this. However, if they don't affect the same gene, you're going to see complementation. Complementation just means that if they're different genes, if this orange gene is different from this purple gene, then the worms here that are mutant for the orange gene are going to have a wild type copy of the purple gene. And these worms here that are mutant for the purple gene are going to have a wild type version of the orange gene. So if you mate them together, those wild type versions of those genes, those wild type alleles, are going to complement the mutant versions, so that together there's a complementary set of wild type alleles resulting in a wild type phenotype, because this worm is heterozygous for both alleles. So if they do complement, that means there are 2 different genes. But if they don't complement, you're going to see that mutant phenotype, and that means that it's the same gene. So I would have loved to try this with HD145. We'll see this later in a different context, but there was no mutant allele available for a candidate region. Very frustrating. So next is a rescue experiment. If we don't have another allele of our candidate gene, then we're going to try just injecting the healthy wild-type DNA straight into the mutant. So this is our mutant worm. Here's our cloned wild-type healthy DNA. And we're going to inject that in. And when it gets into these cells, it's going to create an extra chromosomal array. The blue region is our DNA, and the green region is DNA for green fluorescent protein, so that the worms that have successfully incorporated the extra chromosomal array, or mini chromosome, they'll shine green. Super simple. So our host DNA, the worm itself, is homozygous mutant for our mutated allele. But we've injected this wild type DNA extra chromosomal chromosome. So this is going to be producing healthy protein that this can't produce, resulting in a worm that has a healthy phenotype, even though it's homozygous for our mutant, assuming that these are actually the same genes. So this is a way to test if the DNA you're injecting into the worm is the DNA that's mutated in your mutant. So we would have loved to do this. We did do this. We did this for months, and nobody survived. There was no surviving offspring. Apparently, this stretch of DNA is toxic when there's extra. Oops. So that's life. Moving on to pathway confirmation. In this, we don't actually confirm the identity of our allele. We just kind of use it as a jumping off point, so to speak. In this specific example, our mystery DNA region actually has a human analogous gene, PIGX. PIGX is part of the pathway that attaches GPI anchors to proteins. I'm so sorry, my dear. We're going to make you remember GPI. Luckily, I'm not making you remember, oh geez, what's it? G-protein phosphatidylinositol anchors. Luckily, you don't have to remember that, just GPI anchors to proteins. GPI anchors are what they sound like. They're just a specific type of anchor that attaches a protein to the membrane. And it is assembled in stages with a bazillion different enzymes. One of which is PIGX, and another of which is PIGA. If HD145, our mutation, is an allele of PIGX, then if you mutate a different gene in the same pathway, it should result in the same phenotype, because it's messing with the same process. It should have the same results, right? And also, it should interact with our mutant and other genes in this pathway in predictable ways. We use PIGA1 because we could find it. We found a null allele of PIGA1. We ordered worms that had this null allele off of the internet, and we put them in a nitrogen null background. Essentially, we took out that nitrogen. Remember, all the worms we're working with have no functional nitrogen. So we did that for PIGA1, and these are the results. So let's look at this graph. I'm going to orient you to this. You're going to see a bunch of these, but here's our background normal mutation, just with nitrogen 1 mutated. You notice it's 20 and not 12. That's because the fluorescent protein that we use to visualize the AVG axon causes some extra problems in AVG development, so we're seeing more defects than we were expecting. But that's our background level, so I'm just showing you our background level. Our HD145 mutant has 30% above that. So this is 20, this is 50. So our allele contributes a 30% increase in defects. And so does PIGA1, which is exciting. What does it tell us, though? Does it tell us that HD145 is in fact an allele of PIGX? Does it tell us that PIGA1 plays a role in AVG axon guidance? Does it tell us that PIGX plays a role in AVG axon guidance? Or does it tell us all of the above? Pause. Come right back. You're back? We're back. This is the answer. It does not tell us anything about PIGX. We didn't look at PIGX. So it doesn't tell us if HD145 is an allele of PIGX. It doesn't tell us what role PIGX might play in AVG axon guidance. We didn't look at PIGX. We looked at PIGA1. PIGA1. And it does tell us that PIGA1 plays a role in AVG axon guidance, because when you take it out, we see an increase in defects. What this also tells us is that GPI-anchored proteins are involved in AVG axon guidance. That's cool. That's an exciting connection that people haven't made before, and it's a jumping off place. Because we now know that GPI-anchored proteins are involved in this process, we're not going to come back to PIGX. We're not even going to come back to HD145. We're going to take this GPI-anchored theory and run with it. Because the much more exciting question is, which GPI-anchored proteins are we talking about? What's our question mark here? Our next step is to make nitrogen mutants with no alleles of a bunch of different GPI proteins and look at their AVGs to see what's defective. Theoretically, we would start from the top. Unfortunately, it's a really, really high top. There are hundreds of these, and that's a lot of work. So we're going to try to limit them down. Specifically, we're going to limit them to genes that are expressed while AVG is extending. You remember the video with the blue AVG cell and the teeny little axon extending in the little hook, and that only happened in a tiny little window of time throughout the development of the worm. We're going to only look at genes that are expressed during that time, which makes sense. If it's not expressed during that time, it can't affect AVG axon extension. And there are only 43 of those. Awesome. We're going to prioritize known AVG axon guidance, and we're going to prioritize ones that have easily accessible null mutants. So, at this point in my research, we had been injecting worms like, 10 a week for 3 months, and nothing was surviving, and it was getting really frustrating. And we made this connection of, okay, now we have a list of 43, where do we start? And 4 of them are IgCams. So I reach out to my advisor, and I say, remember that paper that you published in 2009 all about IgCams? I have 4 IgCams that are GPI anchored. Do we already have some, like, in deep freeze? And we did. They were in a giant tub of liquid nitrogen, and I pulled them out and defrosted them, and they grew and were happy. So, let's talk a little bit about IgCams. Also, heads up, my face is about to go away. It'll be right back, I promise. Woo! IgCams are a well-known family of membrane-bound guidance factors. You've seen them before. DCC, which is a netrin receptor, is an IgCam. Robo, which is a slit receptor, is an IgCam. And these 4 are also IgCams, but instead of having these TM transmembrane pieces, these 4 have these little GPI connective regions that get anchored by different enzymes, including PIGA1. So we made those mutants. These are worms where nitrogen is missing, and each IgCam is also missing of the 4. So in this worm, we've seen this before, that was the last table we saw. In this worm, we see, so nitrogen is missing, and Rig3 is our null allele, so these are missing from these worms. They're mutants. So these are Rig5 nitrogen 1 mutants, these are Rig6 nitrogen 1 mutants, and these are Work1 nitrogen 1 mutants. So, based on these numbers, which are the amount to which the AVG axon was misguided, what does this table tell us? A, that Rig5 and Rig6 and Work1 are involved in AVG axon guidance, but Rig3 is not. B, that Rig3 is involved in AVG axon guidance, but Rig5, Rig6, and Work1 are not. Or C, this is actually not enough information, and when this question disappears, there will be more graph. What do you think? Pause it, think, come up with a decision, and come back. We're back! The answer is A. It was not a trick question. Because these 3 mutants show an increased level of defect, we can say that adding these mutations, or removing these genes, either one is correct, results in increased AVG axon defect. But that's not true for Rig3, which looks the same as nitrogen on its own. So we're not adding any new defects here. Okay, so the next logical question is, are these 3 genes working together? Are we getting essentially 3 different pieces of the same legs? Or do we have 3 different unique legs, 3 different ways to keep AVG in the correct place? And the way we answer that question is by figuring out if the phenotypes are interacting or additive. So in interacting genes, it's like each gene is a different part of the same leg. If you take away one, the leg doesn't work. If you take away the other, the leg doesn't work. If you take away both, leg still doesn't work. It's not going to add to the defects. See that? So if they were separate legs, then we'd have an additive phenotype. So we would be taking away both of them, and removing each one is bad, and removing them both is twice as bad. This specific type of genetic interaction is called recessive epistasis. Recessive epistasis is a word I want you to understand. It is when the function of two genes is required for a process. And so loss of both of the genes looks just like the more severe single mutant phenotype. So with this in mind, and it's really important that you understand this, so if you don't, pause, study this, try and make sure you understand why this phenomenon is 40%, whereas this phenomenon is 70%, then come back. Okay? And then we're going to move on and ask a question with this example. Here it is. This graph is color-coded. It's more of the last graph that you saw. In this mutant, there are 3 genes missing. Nitogen is missing. RIG6, the yellow one, is missing. And RIG5, the red one, is missing. So together, they make orange. In this one, RIG5 and WRK1 are missing. RIG5 is red, WRK1 is blue. Together, they make purple. In this one, RIG6 and WRK1 are missing. RIG6 and WRK1, the yellow and the blue, make green. And in this one, everybody's missing. And it's brown, because, you mix everything together, it's brown. These bars show us significant differences. So RIG5 is significantly different from RIG5 WRK1, but RIG5 RIG6 is not significantly different from RIG5. So let's look at these options. A, RIG5 and RIG6 are interacting. B, RIG6 and WRK1 are interacting. And C, none of them interact, or D, all of them interact. Now again, pause and come back. Okay, you're back. Awesome. Ready for the answer? RIG5 and RIG6 interact. And we know that because RIG5 and RIG6 are not together. When you have them together, they're not significantly statistically different from each one on its own. And that's what we were looking for that interaction. Whereas if you look at WRK1 and RIG6, this is an additive phenotype, where you see RIG6 is 20% above Nitogen 1 on its own. WRK1 is 20% above Nitogen 1 on its own. And together, they're 60% above Nitogen 1 on its own. And another thing that emphasizes this fact is that when you add them all together, you don't get higher than 60%. We're seeing 3 20% effect rates. One is from Nitogen, one is from WRK1, and one is from RIG5 and RIG6 together. If you add that all up, we have 3 legs, essentially. A Nitogen leg, a RIG5, RIG6 leg, and a WRK1 leg. Cool? Cool. So, RIG5 and RIG6 interact, and WRK1 doesn't work with them. Ta-da! So now we're going to talk about a couple more mutants that I worked with. And so you'll see how complementation and rescue experiments actually do work. So let's talk about a rescue experiment that worked. So AEX3 was our candidate gene. It was a candidate for the allele HD148. Reminder, things with numbers in them you do not have to remember. We injected the HD148 homozygous mutants with wild-type healthy AEX3 DNA. And these were our results. So here's our control to compare to, again. Here is our homozygous mutant, and here is our homozygous mutant with that extra wild-type purple AEX3 DNA as little squiggles floating around in here. So, what does this graph tell us? A, the wild-type DNA rescued the mutant phenotype. B, HD148 is in fact an allele of AEX3. C, AEX3 is involved in AVG axon guidance. Or D, all of the above. So pause it and come back. Are you back? You're back. It's all of the above. Just this one experiment, this one graph, tells us all these things. And yes, you don't see the word AEX3 in this graph. So, how did we test if AEX3 is involved in AVG axon guidance? Well, we successfully showed that HD148 is an allele of AEX3. So that means that this is an AEX3 mutant right here. We saw it. Here's an AEX3 mutant. It's clearly defective in its AVG axon guidance. So, what does AEX3 do? Full disclaimer, I did not do this experiment. This was my predecessor, Jafar Bott. He looked at AEX3 fairly thoroughly with genetic interaction studies and was able to find out this lovely pathway. AEX3 regulates embryonic vesicle transport, vesicles, little things containing other things. And Jafar was also able to connect AEX3 to ONC5, which is a neutrin receptor. So that's cool. Next! Here's another one. Complementation testing that worked. CCD1 is the candidate gene for HC152, which is one of our mutant alleles. And we were able to get a known null CCD1 allele and cross it with HD152. These are our results. Here's the known CCD1 null allele. Here's our allele. Now, it's not the exact same intensity, but it's the same basic phenotype. We're still just looking at AVG crossing the midline. And this is the F1 offspring of these two lines. So this is not a double mutant. It's one of these two. Which one? What does this graph tell us? A, that HD152 and CCD1 genetically interact. B, that HD152 is in fact an allele of CCD1. C, a double mutant for HD152 and CCD1 has a lower AVG defect rate than an HD152 single mutant. Or D, all of the above. Pause, think about it, come back. Are you back? Yay! This graph tells us that HD152 is an allele of CCD1 because we are seeing defective AVG axon guidance in this scenario. So that means that this guy is a mutant. It's this one. And again, genetic interaction is something that you figure out with double mutants. This is not a double mutant, especially because HD152 and CCD1 are the same thing. If HD152 is an allele of CCD1, then both of these worms have the same have different types of defectiveness of the same gene. Clearly this is less severe than this. So it's kind of fitting that this is in between, even though none of that difference is significant. So, what does it do? Well, nobody knows, but the human homolog version of it does exist. And it interacts with a neural kinase CDK5, specifically in the context of breast cell cancer cell migration, which doesn't seem terribly well connected, but it's a lead. We're going to follow it. Maybe CDK5 interacts genetically, shares pathway, and is part of the same leg as CCD1. We did a genetic interaction study. Here are our single mutants. Here's CCD1 and nitrogen-1 null mutants. And here's a CDK5 nitrogen-1 null mutant. And here is all three of them missing together. Does CDK5 share a pathway with CCD1? Yes, no, or this is a trick and you're going to give us more information soon. All right, you're back. It's cool. The answer is yes, it's not a trick. Awesome. So, in mammals, CDK5 is involved in synaptic vesicle exocytosis. We have seen synaptic vesicle stuff before. Maybe it's part of the AEX3 thing. You gotta check. What do you think? In all of these, we have just the part of AEX3 pathway, and then the AEX3 pathway plus the CCD1 pathway. So, are these all parts of the same pathway? Is this all one leg? Yes or no? Pause. Ready? Yes. Yes, it is. And we know this because we're not seeing any additive phenotypes. Nothing's above 50, really. So all of this is interacting. And I get to add a little leg to the AEX3 pathway. Very exciting. But what about the IgCAMs? Is this also part of the same thing? I mean, maybe work 1 is part of this thing and Rig5 and Rig6 are not, or Rig5 and Rig6 are part of this thing and work 1 is not. We don't know. Let's find out. So I ran this experiment. It was originally more of an I'm bored, let's see what I can do, I wonder what this will look like situation. I wasn't expecting anything exciting. I made a worm that was missing CCD1, missing Rig5, and missing NID1. So it was null for all 3 of these genes. None of these genes work in these worms. Here are the individual lines. So I'm not showing you this worm. I'm showing you the Rig5 on its own and the CCD1 on its own. And I'm going to remind you of what it looks like when genes interact versus what it looks like when it's additive. I'm going to ask you, what do you think this defect rate will be? What will this final bar be? Will it look like a Rig5 mutant? Will it look like a CCD1 mutant? Or will it look like these two added together? Are you ready? Are you ready? This is what I predicted. I predicted that I would see additive interactions, that CCD1 and Rig5 have nothing to do with each other, and I would see a nice big tall line. This is what I actually saw. Surprise! So this is a weird phenomenon called suppressive epistasis. It's when a double mutant looks like the healthier of the 2 single mutants. This is weird, you guys. It works through a regulatory relationship. In this situation, CCD1 is regulating Rig5. So, if you imagine CCD1 as the babysitter to a savant toddler, very intelligent, very productive, and if the babysitter keeps it on task, it'll do great things. If the baby's not there, the toddler is not there, the babysitter has nothing to do, and no great things happen. And that is our scenario for Rig5. So remember, CCD1 is our babysitter, Rig5 is our toddler. If there's no toddler, nothing fancy is going to happen. But if there's no babysitter, we have a rambunctious toddler who's not being directed anywhere. And this toddler is going to run around causing extra problems. So like our toddler, if CCD1 is missing, it can't direct Rig5 to do productive things. So Rig5 is going to do actively destructive things, causing more misguidance and more defects than it would have if you just removed them both. In this scenario, removing both the babysitter and the toddler is just like removing just the toddler. Nothing exciting is going to happen, but nothing destructive is going to happen either. So that is how suppressive epistasis works. And here is a graphic explanation of suppressive epistasis that you are welcome to sit and stare at for as long as you like. This is bonus info. You will not be tested on the nitty-gritty details of how this works. But if you're curious, please feel free to stare blankly at this screen. So, it's time for the summary slide. Today, we learned how to conduct a genetic screen and an enhancement screen. We learned about redundancy in our multiple spider legs and how it complicates our research. We learned about back-crossing and gene mapping and talked about ways to isolate a mutation so that we could figure out where it is. And we learned 3 different ways to identify a candidate gene. Though secretly one of them doesn't identify the candidate gene, it instead identifies pathways. We talked about IgCAMs and GPI anchors. We talked about recessive and suppressive epistasis. And through it all, I've been showing you how to ask testable questions to find testable answers that lead to more questions that you can test, which is my favorite part. Here are your vocab words. Aren't they lovely? You are responsible for these words. I am going to write questions, and then you can answer them as long as you know the meanings of these words. Well, I have had a blast. I hope you've enjoyed this, and good luck. Stay safe, take care of yourselves and each other from a distance. Bye.